Hello students, today's topic is toxicity studies in preclinical trials for drug discovery process. In my last session, I stopped at this stage. The drug development process involves two stages, the preclinical stage and the clinical stage. The preclinical stage includes in vivo and in vitro tests using animal models for studying toxicity, ADME studies, and therapeutic doses. Importance of toxicity study in drug development study is to establish the dose response curve for the lead molecule, to ensure the safety of the lead molecule as a drug before it is registered as a drug for general use, and to establish the mode of action and mechanism of toxic effect if it has been observed in other studies. Three types of toxicity studies are there: acute, subacute, and chronic. Acute toxicity study is the first step to screen the toxicity of the molecule. It is done to establish a dose response curve to estimate the LD50 value, which is expressed as mg per kg of body mass, to study the adverse effects of the drug. In the experiment, the biological effect of the molecule is also studied. Example, time of onset of response, duration and degree of recovery on the survival animals. The animals are monitored and the behavioral pattern and other movement or symptoms are recorded. Example, motor activity, anesthesia, tremors, arching and rolling, clonic convulsions, which means jerking the body or body parts, lacrimation, meaning flow of tears, storm reaction, meaning the animal carries its tail at a vertical position, salivation, muscle swarms, withing, etc. The different methods used to evaluate acute toxicity is arithmetical method of Carber, graphical method, arithmetic method of Reed and Munich, and Lorke's method. We'll just study two in detail. First, the arithmetical method of Carber. This is the simplest and a crude method and is performed on a small group of animals divided into 5 groups, generally with 10 individuals in each group. Each group is given a specific dose of the molecule or substance to be tested, with increment in dose from group to group. Here, the difference between the doses given to each group is a key parameter for calculation of the LD50 value. The interval mean of number of mortality is recorded for each group, and the lethal dose LD50 is calculated by the formula given below. The results from doses larger than the lethal dose and smaller than the minimum dose are excluded from the study. Let's take an example for determination of LD by Carber's method. The table below shows the experimental results. The animals were divided into 5 groups. Each group had 10 individuals. The dosage given to the first group was 64 mg per kg body mass. For second, 71 mg of the substance per kg body mass. Third, 81, fourth 90, and the fifth group received 100 mg per kg body mass. In the fourth column, the dose difference is calculated, which is represented as letter A. That is, the second row, 71 minus 64, the dose difference is 7. 81 minus 71, the dose difference is 10, and followed by 9 and 10. The mortality rate of the animals were counted and recorded. For 64 mg per kg, there was no mortality. For 71, 2, 81, 4, for 90 mg per kg, 9, and for 100 mg per kg, 10. The mean mortality rate was then calculated, which is represented by B. The last column is the product of A and B, that is 7, 30, 58.5 and 95. The sum total comes to be 190.5. The LD50 value is calculated by the LD100, that is 100 minus 190.5 divided by total number of individuals in each group, that is N. This comes to be 81 mg per kg of the body mass. Next, graphical method of Miller and Tainter. In this, similar to the last method, a small group of animals are divided into 5 groups of 10 animals per group. Each group is administered with different doses of test substances in an increased way or incremental way. The log doses are plotted on a graph against probits, which was proposed by Bliss. This is then used to calculate the LD50 value. The method of calculating LD50 value in graphical method. First, by hit and trial method, the least tolerant dose and the most tolerant dose is estimated. Then 5 doses between the least tolerant and the most tolerant doses are taken, and these are given to the groups. After tabulating the results, apply the correction factor to 0% and 100% mortality groups. The percent mortality values are then converted to probit values by reading the corresponding probit unit from the probit table. Plot the probit value against log of doses and estimate the LD50 value. Example for calculation of LD50 value in case of graphical method. The first table on the left shows the results obtained from an experiment. 5 groups, each group containing 10 individuals. The dosage given is 25 mg per kg of body mass, 50, 75, 100 and 150. Next column, the log of these doses are calculated. Followed, the next column shows the percentage of dead in each group after the given dose. Then the corrected percentage is calculated. The percentage of dead for 0 and 100 are corrected as shown in the figure on your right top. Then for each value, the Forbit value is estimated by the Forbit table that is shown in the next slide. After calculation of the Forbits for each dose percentage, the table is created or made against log of doses versus Probit. The x-axis is represented by log of doses and the y-axis the Probits. From the table, the LD50 value is estimated. Example, for here the LD50 value or the log LD50 value is 1.76. Antilog of 1.76 is 57.54 mg per kg of mass. The table here is the percentage mortalities to Probit or the Probit table. Considering the earlier example here, percentage of mortality 40% corresponds to 4.75. 70% mortality corresponds to 5.52 and 90% corresponds to 6.28 Probit value. The arithmetical method of Reed and Munich, which was proposed in 1938 to determine the medium lethal dose. In this analysis, cumulative analysis of the values are done. The cumulative dead and the survivors are recorded. Doses larger than the least tolerant dose and smaller than the most tolerant dose are eliminated and the LD50 value is calculated. The Lorca's method of acute toxicity test. In this method, the experiment is divided into two phases, phase 1 and phase 2. In phase 1, 9 mice are utilized. These are divided into group of 3, each containing 3 individuals. Each group is given different dosage of the test substance, example 10, 100 and 1000 mg per kg. After the administration of the dosage, the mice are monitored for a period of 24 hours and the results are recorded. Next, in phase 2, it involves 3 animals divided into 3 groups, that is, each group containing only 1 animal. The dose level chosen could be either step up or step down depending upon the results obtained in phase 1. The toxic symptoms are observed for 24 hours as well as delayed toxic symptoms are monitored for 7 to 14 days. The lethal dose is calculated by the formula given below. The table here shows the calculation of LD50 value by Lorck's method. Subacute toxicity study in preclinical trials. This is conducted to determine the organs affected by different dose levels. The dosage level is calculated based on the information obtained from acute toxicity studies. Generally, 3 doses are given: high dose, intermediate dose, and low dose. The results are extrapolated using the area under the curve and Cmax. We had discussed about these in detail in my earlier session. The LD50 value is then calculated. Chronic toxicity study in preclinical trials. This is to determine the organs affected and to check whether the drug is a potential carcinogen or not. It involves a large group of animals and extends for a period of very long duration. I had discussed about this in my earlier session in detail. Presently, there are alternative methods too for the clinical studies. Example, genomics, proteomics, and metabolics, plus use of certain transgenic animals and cell culture lines. These two I had discussed while discussing alternatives to animal cell in toxicology. ADME studies in preclinical trials. The assessment of pharmacological properties ADME is important for any selection of lead components. The flowchart here shows the two-tier approach of in vitro and in vivo analysis for ADME studies in preclinical trials. In the first two tiers of ADME analysis, in vitro studies are carried to study the lipophilic, the solubility, and the microsome and plasma stability, plasma protein binding, screening of cytotoxicity and hepatotoxicity, cytochrome 450 inhibition profile, and permeability of the target compound. The lipophilicity test is done to calculate the log D value or the coefficient of distribution. It is measured by partitioning it against octanol and the buffer at 1:1 ratio. It plays an important role in drug solubility, absorption, membrane permeability, plasma protein binding, and distribution. Solubility is estimated by dissolving the test substance in various buffers at varying pH. The microsome stability is estimated using liver microsomes. The test sample is incubated in microsomes and the liver microsome contains the various metabolizing enzymes like the monooxygenases and carboxyesterases. The microsome stability is then checked by LC-MS. The measurement is done at specific time intervals. The screening of cytotoxicity and hepatotoxicity is done by using specific cell lines. This is a list of certain medical terms used in the present session for your referral. Thank you.